Hey there.
How's it going? How are you?
Doing well. Nice haircut.
Thank you. Summertime.
Yep. Yeah, you lived in Miami, right?
Yeah, yeah. It's nice and warm here. It's like
Yeah.
sometimes it feels like 104 some days, so
Mhm.
it's hot.
Yep.
How was your weekend?
It was good.
Nice. How's New York in summer?
It gets hot and humid here. It gets
hmm
pretty hot and humid here, yeah.
Yeah, Miami's pretty much the same, but I guess I have some friends from New York and I was with them a couple weeks ago. They were saying that Miami's nice because it's open air. It's like not like a concrete jungle, right? Like
Yeah, yeah, yeah. Yeah.
it's um sorry, I'm just turning my iPad on. So, um, I think tomorrow is the day that I take the blueprint for Altius.
Yep.
Yeah.
Yeah. How do you feel?
Uh, I feel good. I know there's 5 weeks and change till my exam. Um, I'm not sure. So I've decided, just to give you an overview, I've decided to wait to apply for next year. Just because it was all going to be a bit of a rush. I was still struggling to get my letters of recommendation and finish all my activities and stuff. So I figured it'd probably be best to wait. Um, Also get I only started really trying to do this a year and a half ago. So I'm still lacking some sort of clinical. I have clinical experience, but not really in depth enough.
Yeah.
Um, so I'm going to give it a year and try to, improve in every little area, get a little bit more volunteering and stuff.
Mhm.
Uh, so I'm trying, so I guess there's 5 weeks. My thing now is like I have all the knowledge in a sense. Like I studied a good amount. I suppose taking the test July 13th is probably the best option. But at the same time, because I have a year, then I'm also considering, oh, maybe I leave it till like the end, right? Like September or something. And then study,
Mhm.
likely over the summer, a couple hours a day. Um, so I don't know, maybe to see what you think.
Yeah, I think that's a good idea. You could take it on the fifth. So I think you're taking it on the 15th, right? Rather than
Yeah, I think it's on the 13th of July. Yeah, so it's a Saturday.
okay.
Yeah.
But yeah, you could take that exam, do your best, and and then you can, get your result back, but you could then continue studying and then taking the test at in September would be a good
Yep.
bet.
Okay. Oh, you think that would even then that would be okay to
Mhm.
do it 3 times?
To do it how many times? 3 times, you mean?
This would be my second time.
Ah, got it. Okay. What are they? Yeah, I'm not really aware of what they say typically about taking it like however many times. Like, what do they say that it's bad that if you take it 3 times?
I don't know if they say that it's bad. I can ask
Yeah.
and see if they, I suppose like the GMAT and the LSAT, there's really no repercussion for doing it a bunch of times.
Yeah.
But also I know that you can see your score on those and like cancel it basically if it's not the score that you want.
Yeah. Let's see.
Yeah, I mean, I guess I
Okay.
suppose it doesn't matter too much.
Okay. So you know what? Um, but I was just like Googling it. Let's have you. So yeah, my initial thoughts were that you can go all out because like if you push it back to like September, it will feel like you know um that it's so far away that you may not put in the same level of work.
Yeah.
So my thoughts were like to take it on in July and going in like or going in and doing as the best you can. But also knowing that you can take it again in September. But I looked it up and it seems like, you could take it 17 or rather 7 times in your life, three
Hmm.
times a single year. But most people are saying to limit it to 3 times.
Yeah.
So I think that means that, yeah, it would be better for you to not take it in July and take it like in September.
Hmm.
So, and worst case scenario, you don't get the grade you want in September. If you're Since you're applying next year, you essentially have until like let's say June 1st
Yeah.
of 2025.
Yeah. Ideally, I mean, I imagine my only worry, not worry really, is just um it feels almost as if like I spent all that time studying and then I wait
Hmm.
like a waste. I know it's not a waste because the information was learned. I guess the skills
Hmm.
don't really get lost if I apply them, maybe a couple hours a day, maybe a couple hours a week. Um, but obviously you're the expert. So if you think that it's a better thing to do, and really there's no rush since like you say, it's till June 1st. Uh, I'd like to do it, the best way possible.
Yeah.
Um,
Yeah. So I would say September,
mhm.
max amount of, for this year, the max amount of time will be, if you take it in September, and then the maximum amount of time would be the June, early June of next year. So that would be 3 times in total. So
Mhm.
it meets that kind of thing that med schools look for. And
Yeah,
also benefit from having as much time as humanly possible. So
yeah.
yeah, I think that would work.
Okay. And say that I move it to for the September date. Um, how many hours do you suggest I work a day? I mean, obviously, like I think if I'm gonna wait till then, I'll probably try to get like a part-time job or something. Maybe 20 hours a week. How many hours would you suggest would be the best to study till then?
Um, so okay, what's like a target score for you?
Um, I want to be like at above a 515, I guess. Like, um, I know that I could get within that range, so I'd like, yeah, anything above that, really.
How many hours are you putting in a day today? I mean, like,
Yeah, it's 5 hours, maybe 5, 6 hours. Before I was putting a lot more, maybe
yeah,
not efficiently. But 5 hours, yeah, 5, 4 hours. I do like an hour and a half of Anki, like 60, 80 questions of UWorld, and then some content review, and then cars, obviously.
got it. Okay. So, um, yeah, so okay, so are you asking like how many hours you're gonna do, like if you take it in September or something?
Yeah, I mean, just to see, because like I suppose I wouldn't be following the same schedule as the one here. Like I wouldn't have to condense a full length every 3 days. Um,
Yeah.
and then maybe I can spend more time reviewing a full length instead, like maybe do one every couple weeks.
Let's do a full length every week, and because I have it down as 2 per week,
Okay.
but let's have you do 1 per week.
Hmm.
But um, but yeah, let's make sure we finish. Like, oh yeah, I didn't ask if we finished the AMC section bank yet or not.
No, I haven't. I've been doing UWorld mostly since I had done the section bank. Um,
Crap. Okay, okay. I
yeah.
keep forgetting that. Okay, so yeah, because in my notes here, I wrote section bank check mark, but in the spreadsheet, I wrote section bank. So
Oh, okay,
that,
yeah. So I've
that
just been doing UWorld.
Yeah, yeah. So okay, let's go from 2 tests a week to 1 test a week,
Mhm.
and everything else in between will be UWorld. And now that we have this much time, let's aim to actually finish UWorld, like all the questions.
Okay.
Not their cars questions, but all the other questions.
Mhm.
So that usually works for a lot of people. Like I've never, come close to like, finishing UWorld, but I've had some students in the past who did finish UWorld and they did really well. But you also want to make sure that you're doing like, your cars stuff
Yeah.
every day. Um, so yeah, so okay. I mean, like I can adjust this schedule for you. Uh, but what I would adjust is just 1 test a week.
Mhm.
Everything in between would be UWorld and either AMC or Jack Weston cars.
Mhm.
That should provide you with enough resources. So yeah, I think that would.
Okay. So you think that's a better option then?
Hmm.
Okay. Should I take the full length tomorrow, see how I do, and then gauge from there just to determine whether or not, or should I just try to reschedule till then?
Yeah, I would say yeah, if you could take it tomorrow or you know like let's say Friday or Saturday or something like that, that would work.
Yeah.
Yeah. Because yeah, at the end of the day, I still think the most important thing to like help your score is just to take as many full lengths as possible.
Mhm.
So, um, so yeah, I think by September, we'll have finished, or you'll have finished all like, well, maybe not by September, but because I have to do the math here, but I think we can get you to complete all 10
Mhm.
a blueprint or LTS exam to give you like that plus finishing UWorld. I think that will put you in a good position.
Okay, perfect.
Hmm.
All right, that's good. Then I'll take the test tomorrow. I'll let you know how it goes, what score, or maybe Thursday.
Mhm.
Um, and then I suppose, yeah. So if I reschedule before the 12th, I don't have to pay like the $150 or whatever to to reschedule.
Yeah. Yeah. So yeah, for the test, it's your choice to whether to take it like tomorrow or Thursday or Friday or Saturday. Up to you,
Mhm.
whatever feels right for you. Um, and are there any like problem areas content wise that that you have that maybe you can do like let's say you have some issues with like whatever like HNMR or something like that like whatever those problem areas are you could kind of just do UWorld stuff for that like for this week and then take your test at the end of this week.
Okay.
That could be something, or you can take the test tomorrow and then find out what types of things to go over.
Mhm.
So yep, it's up to you. I would say that you could take it anytime this week,
Yeah.
and from now on we're going to just do like you know 1 test a week. So
Okay.
yep.
Sounds good. Great. All right. I'm glad that that you approve. I
Yeah, because I
feel like it's more reasonable.
what's up?
I feel like it's more reasonable to give if I have more time, then why not take advantage of it? And
Yeah, no, exactly. Like that's the toughest thing about like most of my students is like they're they're like applying, so you know their test score was expected like a month ago or something. So uh yeah, it's really hard to set that up right. But you're doing what I would do, which is choose whatever date gives me the maximum amount of time.
yeah, okay.
So,
Nice.
yeah.
Sweet. All right. I guess I will do a little bit of review then. I started doing the Panko deck.
Yeah,
Uh,
I hear
it's
that that's like so.
super helpful. Yeah. It's a lot, but it's I guess like now that I have time, then
Mhm.
it'll be good to bring my score up.
Yep. Okay. So yeah, what have you been doing? Oh yeah, you've been doing UWorld stuff.
Yeah, I've been you rolled sort of like every day, for example, like yesterday I did um I did physics, and then I'll do a little bit of content review on physics stuff.
Mhm.
Uh, what else have I been doing? Biochem. I've been doing quite a bit of. I was struggling with some of like the protein stuff or like the structures and whatnot. I was just reviewing a bunch of that. Enzymes, I felt like I was doing pretty well on those, like the kinematics questions.
Mhm.
Uh, but that's pretty much it. That Anki and then cars every day, two passages. And cars has been getting a lot better. I feel like it sort of depends. Some passages I know that I'll do a lot better on because I can understand them, and then some maybe they're a little bit more verbose or
Are you saying the ones that you understand better are like the social sciences ones?
yeah, for the most part. Yeah, social sciences ones I prefer. The ones that the philosophy ones or like the art ones sometimes are a little abstract
Yeah.
and they like almost take me off the path and then I have to circle back and then it's just wasted time and so that's something that I've been working on as well.
Yeah. Yeah, those types of passages could be that's those types of passages are usually tough tough for people. I used to do something, I don't do it anymore just because it's like consuming, but like if you like click on, the cards question pack, you can have it show like which topic
Mhm.
type passages that you are doing well in and doing poorly in, and I would make like a spreadsheet which would rank them. So like, like let's say like art, history, literature, philosophy, like in order of like least comfortable to most comfortable. Like I would do something like that. But that's kind of like, well, time consuming and at the end of the day, you want to be able to tackle any type of cars passage. So um but yeah, if you do recognize that the ones that you're having trouble with are the more abstract, like art, lit,
Yeah.
philosophy, yeah, those could be good, yeah, to practice. Um yeah, how's cars going for you, like the AMC stuff? Or
I've been doing one AMC, one Jack Weston.
yeah, because you did the two cars question packs, right?
Yeah, so I'm still finishing the diagnostic.
Yeah,
Uh,
got it, got it. Okay, awesome, awesome. All right, what would you like to do
I
today?
was I was thinking maybe we can do some biochem, maybe like I
Mhm.
don't know, 45 minutes and then 45 minutes one passage if you
Yep.
have time. We have till 4:30, so an hour and a half.
Mm-hmm.
All right. Should we do UWorld or AMC?
Um, what type of BP topic?
Um, maybe I don't know. I'm trying to think what would be best to look at with you. Um, something maybe I don't understand too well. Could be like um maybe then something that I don't do too well on is like cellular biology. I always have trouble with those.
What
Uh,
what what biology?
cellular biology, right? What would that be? Like genetics or like um, DNA technology, like PCR techniques and
I
recombinant DNA, all that stuff. I don't struggle with understanding. Maybe just like the application in passages is tough.
okay. So yeah, let's do some UWorld maybe.
Okay. All right.
Um yeah.
All right, how many questions should I pop up?
Um, let's see. Yeah, see if you could share your screen so we can see or decide what to select. You said cellular. Oh, okay, you already have that selected. That's
I guess we can do a mix up of things. Doesn't have to be that specifically, but
yeah, what else would be something?
um maybe molecular bio as well. Just like throw that into the mix.
So let's see. Hmm. Okay, let's have you just let's have you just select cellular biology,
Mhm.
and then you can make like number of questions like 55.
Okay.
Yeah, I think generally for this, whenever you make these types of things, like making it like 59 questions, kind of like, it would be on the test.
Okay. All right, I'll generate it.
Okay.
Okay. Um, so for these, I guess I'll just read them out loud. Or
Yep.
which types of DNA are contained in a plasmid that can be used to transform bacteria in an experiment? DNA that is um, so I know that plasmids are typically circular, right? So
Yep.
it would be three only.
Good.
Yeah. Okay. Yeah, that I understand pretty well typically.
Hmm.
Um, so autonomous retrotransposones are segments of DNA that encode specialized enzymes. These enzymes allow autonomous retrotransposomes to move between different regions of a host genome via RNA intermediates using mechanisms similar to those of retroviruses. Some autonomous retrotransposomes are segments of retroviral DNA that have been partially inactivated by mutations. Given this, which of the following mutations would most likely convert active retroviral DNA into an autonomous retrotransposome?
All right, lots of stuff happening in this question stem here. Let's make sure we understand what they're asking us.
So they're asking us based on the mutations that they're giving us, which one would most likely convert the retroviral DNA into essentially like a DNA that can encode for enzymes.
Okay.
And we know that enzymes are typically proteins. So maybe we're looking for a mutation that would convert retroviral DNA into a protein.
Okay.
Um, so a mutation that prevents the expression of functional reverse transcriptase. So this would prevent uh, RNA being turned into DNA, basically, right? Into complementary DNA.
You're saying
This
the reverse transcriptase?
Yeah.
Okay.
A mutation that prevents integration of retroviral DNA into the host genome. So active retroviral DNA. So we're looking for something that converts active retroviral DNA into an autonomous retrotransposome. Okay. Sorry, it's a weird question. A mutation that prevents the expression of envelope proteins. Um, this most likely would be something that doesn't convert. um, and a mutation that prevents RNA polymerase from transcribing retroviral DNA. RNA polymerase. Um,
Yeah. So let's see, right? So they're talking about autonomous retrotransposons that seem to be just segments of DNA that code for specialized enzymes. And then these enzymes allow these autonomous retrotransposons to move between different regions of a host genome via RNA intermediates using mechanisms similar to those of retroviruses. So they can um, move between different parts of the host genome, kind of similar to how a retrovirus does that. Some autonomous transposons are segments of retroviral DNA which have been partially inactivated by mutations. So when we talk about mutations here, we're talking about inactive, um, you know, segments of retroviral DNA.
Mhm.
And they're asking us, given this, which of the following mutations would most likely convert active retroviral DNA into an autonomous transposon, right?
Yeah.
So we need to find out um, something that, well, something that would convert the retroviral DNA, the active form, into this autonomous retrotransposon.
Yeah. Which is like a virus, right? Or like a or yeah, I mean, I don't know. They're saying that it's a segment of DNA that encodes for enzymes, but they're also saying it's similar to a retrovirus,
Mhm.
looking at something that would prevent the conversion or that would convert it to an active autonomous retrotransposon from an inactive
Mhm.
retroviral DNA, I guess, right?
Yeah. So okay, this is a pretty tough here. Let's think about if any content knowledge can help us. Like, what would be, well, actually, yeah, I was gonna ask what would a retrotransposon be? But I guess
Mhm.
they kind of describe, what it is.
Yeah.
But what do you think it is?
Man, I know that a transposon essentially can like move a segment of DNA to another point, or a gene to another point on that DNA.
Huh?
So maybe a retrotrans, it means it returns it back to its original form.
Huh? Yep, and it moves, using these RNA intermediates.
Mhm.
So if we think about that with RNA intermediates, that means that that portion of the DNA must be um, transcribed into
Mhm.
the RNA.
Yeah.
And okay, now if the mutation uh, deactivates this, we
Yeah.
want to figure out what would, yeah, what would stop that, or sorry, what would make that retroviral DNA deactivated. So,
What would make it deactivated?
mhm,
Oh, okay. Yeah. Okay. So there would be something that prevents the RNA polymerase from transcribing. Wait, but that's not what RNA polymerase does. Doesn't transcribe retroviral RNA DNA though, right?
mhm.
Um, and we're not talking about expression of proteins. So maybe it's either A or B, maybe, and then the integration of DNA into the host genome. I don't know. B confusing me. I'd probably guess A. But
All right, let's see. So A says a mutation that prevents the expression of functional reverse transcriptase. So if that was the case, we okay, let me ask you this first. What purpose does a virus have in infecting a host cell?
it can essentially replicate itself, right, by using the host machinery.
Okay. So let's say we wanted to stop that from happening.
Then you would need it to not be integrated into the genome,
All right.
that then transcribed into proteins or sorry, RNA and then proteins. So I guess then to stop that, you would want to prevent its integration into the host genome.
Uh-huh. And by extension, maybe prevent integration into the host cell.
Mhm.
So what choice would be, yeah.
Uh, B,
Which one?
B,
You said B? B as in boy?
as in boy.
So let's see. Okay, that prevents integration of the retroviral DNA into the host genome. Um, which, I guess would mean that the viral DNA is not getting integrated, I guess, into the host genome. Um, what choice, though, could make it kind of even more
Okay. Yeah, I
unlikely?
guess preventing reverse transcriptase from converting it to DNA.
Okay, let's see.
But my thing is the question saying which of the following mutations would most likely convert retroviral DNA, but reverse transcriptase works with single-stranded RNA, doesn't it, to make complementary DNA?
Yep, it could do that. So let's just think about, because if you had, the or like the reverse transcriptase, or rather, let's think about it this way. Like, what's the best way to prevent anything involving like a virus, let's say, entering into your host cell? Because we've been talking about how to prevent it perhaps from, entering the nucleus and like having, it get uh transcribed and stuff.
Yeah.
Um, but is there anything we can do to make that so it doesn't cause it to be transcribed, but mm, maybe can work even before that it gets a chance to do
Yeah, I
that?
mean, I guess it could be like, I don't know, taken up by like lysozymes or
Uh-huh.
uh like the cell's defense mechanisms before it even gets close to
Uh-huh.
the nucleus. So
So this is a what?
a bacteriophage.
Yeah. Now that just infects bacteria,
Mhm.
right?
Mhm.
It shouldn't affect us, right? Or animals.
Yeah.
How would it look different for an animal? Or rather, like a virus that would infect an animal?
Um, how would it look different? What's like an enveloped
Um,
virus?
yeah, yeah, right.
Yeah,
'Cause-
so as
yep,
like I don't know, binding sites on the outside of it.
Yeah, right. Like if this is like an animal cell and we have our virus here, when it buds off to give you like to give the virus like a membrane, and since animal cells have that cell membrane, it can just enter in and the membrane will be kind of like, enveloped.
Yeah.
So that could be a way for us to prevent um or to deactivate retroviral DNA
Mhm.
because it's not going to even get a chance to enter that cell,
Yeah.
let alone, the cell's nucleus.
Yeah, that makes sense.
Hmm.
So in that case, we'd go with C, right?
Hmm.
Something that prevents expression of envelope proteins.
Yep.
Nice. Okay. Man, that's a tough question.
It is. It is.
The percent. Oh yeah, 21%.
Oh, yeah, that's lower than all the other ones. Yeah. Yeah, I always think that that means it's a good question because yeah, that's the this
True. I
yeah,
struggle.
that's what
Yeah. And also, I guess you learn the most from questions like this.
yeah, yeah. Yeah, because every other choice, and like I kind of also know this because I've made a lot of MCAT passages and questions. Um, and what you want to aim for if you're making, answer choices are like logical, valid methods. Like, it can't just be like completely like crazy wrong. You want it to
Yeah.
be pretty plausible. So yeah, this is a tough one. I mean, the question stem alone is extremely long and yeah, it's super tough here.
Yeah. Nice.
Mm.
Okay.
Okay, that's a good image right there. Yeah, you
Yeah.
see the envelope, yeah, like gets absorbed into the cell bilayer, and then yeah, it
You're
opens
saying.
up there. Yep.
Okay, so those other ones, I guess, would have been even later. They wouldn't have been the best sort of way to prevent it from.
Yep, yep.
Okay. Nice. Yeah, it's a good picture.
Yeah.
Yeah. Like that. Alrighty.
Did you want to do like something like this, or do you want to do maybe like the discretes first?
Yeah, I guess we could do the discretes first. Yeah.
Okay.
Okay. All right. So the diagram below shows a part of the life cycle of a novel virus that infected an E. coli cell. Which of the following experimental techniques can be used to detect genetic material of this virus in the host cell? So we're looking for an experimental technique that can detect the genetic material of a virus, specifically a bacteriophage.
Yeah, what a coincidence, because I just drew one.
Yeah,
But yeah, okay, yeah. So what's it asking us here?
it's asking us to give them the experimental technique that would be used to detect this virus or the genetic material in this virus. Um, so I suppose that based on the image, that's I mean they don't tell us if it's DNA, RNA, single strand, double stranded.
Okay.
Um, but I guess because the picture of the genetic material being translated into proteins, it's most likely RNA.
Yeah, right. Because you don't have transcription have it happening.
Exactly. So we're using an experimental technique that helps us find RNA. Um, so I think I'm gonna write the mnemonic down because it's just a little bit easier. So it would be northern blotting for RNA.
Yep.
Um, yeah, I guess, right?
Yep.
Yeah, yeah, yeah. And then I think obviously I've heard of like DNA sequencing. Would that be like PCR?
Um, so like DNA sequencing, they have different methods of uh pretty complicated methods of like, like adding nucleotides and using some way to figure out which nucleotides we have. Um, would be the sequencing part. There's like high throughput sequencing and things like that. But for the what was the one that you mentioned just now? The
PCR, right?
Oh yeah, so PCR would be not sequencing like a genome or anything, but it would just amplify some, you know, some DNA or RNA in your body. Well, RNA, I guess, to make it so that it's easier to uh deal with, kind of. It's like all right, like I was watching like a TV show or like a true crime documentary about some crimes, obviously, and then people who left like some saliva or some type of fluid behind in like the 80s and things like that, and they didn't have the technology to really be able to do anything with that. It's that's more of like that's more of like a recent thing,
Yeah.
but because so like a lot of these police officers or whatever, they just kind of save the evidence in they store it or whatever. Um, because prior to that, they would, in order for them to do like a DNA analysis, they would need like that entire, like let's say some bad guy left like a milliliter of something, right? A fluid, a biological fluid. Um, if you wanted to sequence that, it would take the entire sample, but RNA, or rather, yeah, PCR lets you amplify a tiny, tiny segment of DNA or RNA. So that's what that would be.
That's cool.
It's like, yeah. Um, yeah, pretty much that.
Okay, I remember that now. It's easy. It's like a good story to plug it into.
Hmm.
Um, okay. So scientists studying evolution in yeast found that certain species underwent a reduction in chromosome number with no loss of coding information. Given this, which of the following mechanisms best explains how this reduction in chromosome number may have occurred? So we're looking for um, yeast that have had a reduction in chromosome with no loss of coding. Now they're asking us essentially to explain which mechanisms can explain for this. Um, so one, end-to-end fusion of two chromosomes followed by an activation of one centromere on the newly formed chromosome. So um, end-to-end fusion of two chromosomes. So first, they're telling us yeast. Yeast a bacteria. Yeast is bacteria, right? Um,
Um, uh, I think it's like a mold.
a mold, like a fungus.
Yeah, yeah, I looked it up. It's a eukaryote. Yeah, it's a fungus, and
Oh,
it's eukaryotic.
okay, yeah. I don't know why I thought it was bacteria. Um,
Probably because it's super small and infects people. I mean, I wasn't sure if it was the fungi until I just looked it up right now.
um, okay, so I mean, I guess it doesn't really tell us much then. Um, well, end-to-end fusion of two chromosomes, followed by an activation of one centromere on the new on the okay, so I know that the at least content-wise, like the centromere doesn't really have much coding information.
Hmm.
it's like a heterochromatin.
Okay.
So maybe that because it's inactivated doesn't really affect the coding information.
Okay.
And then end-to-end fusion, I mean, I can imagine what it looks like, but I've never read about it, or at least I don't remember.
Mhm.
Um, but I wouldn't imagine like end-to-end fusion seems like the gene sequence continues extending. It doesn't really get cut short. So
Uh-huh.
maybe one works. Um, two saying breakage of a chromosome at the centromere. So I guess the sister chromatids are probably separating and fusion of two resulting chromosomal portions to the ends of the other chromosomes. Uh, so I guess it's saying, well, I guess like if you have like homologous chromosomes, then the sister chromatids separate and maybe swap over.
Okay.
Um, and then three saying transfer of a DNA sequence from one chromosomal arm to a different chromosomal arm. So that the three wouldn't work because it's not a reduction in the chromosome number.
Okay.
It's two either if they're reattaching to the ends of the two breakages, then you wouldn't lose chromosome number. They would just equate to the same. So maybe it's just um A one.
So yeah, sure. If we have something like this, like these two chromosomes,
Mhm.
and add them like end to end,
Yeah.
that, but we can't have two centromeres like this. So one of these leaves or is lost
Mhm.
to give you something like, well,
There's like a super long leg or something. Yeah.
yeah, exactly, something like that. So, you know, this is a reduction in chromosome number, right?
Mhm.
And this isn't missing any like coding information, right? Because that, as you said, the centromere doesn't have much coding information in it, right?
Yeah.
So, yeah, I would say that Roman numeral one makes sense.
Yeah, I just read over two again, and maybe two makes sense as well. I was like trying to picture it right now. So I guess they're saying if you break off the chromosome at the centromere and you're left with each arm, then they're getting those two arms, the two resulting portions, to the ends of other chromosomes. So maybe it would just be like a same thing, basically getting rid of one chromosome, right?
Hmm. Yeah, breakage of a chromosome at the centromere and fusion of the two, uh, resulting chromosomal portions to the ends of the chromosome. So this is essentially what Roman numeral one is saying, except the order is changing, right? Like Roman numeral one says the fusion happens first, followed by the inactivation of a chromosome.
Yeah.
And two is saying that the chromosome is breaking at the centromere
Yeah.
and then fusing.
Yeah.
And so, okay, that works. And I guess that will give you the answer because I didn't like hide, those choices, but let's just think about that for a sec. Roman numeral three, like transfer of a DNA sequence from one chromosomal arm to a different one. What does that sound like, I guess?
So that sounds like almost like a transposon, right? Like
Yep, exactly.
it's moving an allele to another.
Hmm.
So I guess yeah, that wouldn't reduce the chromosome count. It would just move over that sequence to another sequence.
Perfect.
Nice. Okay. Yeah.
Oh yeah,
Nice.
yeah. You could see how we have fusion and then an activation of chromosome. I mean, centromere, but here we have centromere, I guess, breaking and then the fusion.
Yeah. Yeah, that's good.
Mhm.
That makes sense. One second, I'm just gonna let my dog out.
Sure. What type of dog do you have?
I have he's a bit of a mutt. He's like a Dachshund mix. We rescued him, so he's like a super mutt. We don't know really what he is.
I got this one over here.
So cute.
And
Nice.
her
What's it?
necklace is made out of tritium.
Oh, that's sick.
Uh-huh.
It's cool. It's just where you could see her in the dark.
Yep, yep.
It's
So
nice.
yeah, I had to get it. Like, have you ever seen like Spider-Man 2, the old one with like Doc Ock?
Yeah, I think I mean, I haven't seen it in a while, but
Yeah.
yeah, I definitely
He starts his like fusion like experiment with tritium. So like when I was a kid, I learned that word, and I see it a lot on the MCAT as well. So
really.
yeah,
I know I've seen some of your videos talking about tritium.
yeah. Yeah, it's really cool stuff because it essentially just, and yeah, you probably know, like, it will be on like the ends of like your sights for like, certain types of guns and stuff. And
Resin battery.
yeah, exactly. And it's like, yeah, it's really, really cool.
That's cool. I didn't know it sold necklaces.
Yeah. I was like I'm pretty sure that like my cat is the only one that has a tritium necklace like this because I ordered it from this person in Taiwan because apparently they have that like
Yeah.
left over from like their whatever nuclear power plants. So yeah, it's pretty awesome.
What's the like the machine they use at museums to test radiation levels?
Like a Geiger counter.
Counter, have you ever used one on it? On the
No. I remember on like Slick Deals once there was a sale for like a Geiger counter. Uh, but yeah, I never purchased that because um, you know, like what should be happening is that like let's say we have some tritium here and then like out or surrounding it is like a type of phosphor coating that is what, and so the tritium is going to release beta rays that will be taken up by the phosphorus like coating to give you like a particular color.
nice. That's cool.
Yeah.
Oh, okay. So that's how they give the color. They just pick like a type of mineral, I guess, to
Wait, what do you say about that? The type of,
how do they pick the color though? Do they, I don't know, is it like mineral based? Different minerals will shine different colors or
so yeah, it just the phosphor like coating of it.
okay.
That could be like that can like let's say they use a type that when it absorbs the beta radiation, it can produce a certain color, or a certain wavelength. And so they could just use slightly different versions of that coating to give you different types of colors when it glows, essentially.
Nice. That's cool. Nice. I'm gonna look at necklaces to see. They have them on like or something.
Yeah. It's pretty cool.
Nice. I guess let's do these, right? Should I read this? This is yeah, this is like cell bio, right?
Um, yep. Everything should be.
Yeah. Okay. So an epithelial cell in the upper respiratory tract was infected with an enveloped virus, such as influenza virus. Upon entering the host cell, the viral life cycle was initiated, and the host cell machinery was used to synthesize new viral components. Given this information, which of the following statements is least likely to be true? Okay, so they're saying um, once the virus entered the host cell, the life cycle was initiated and the host machinery was used to synthesize. So this seems to me like they're talking about the lysogenic cycle here. Um, maybe. I mean, I guess I can read through the. So which of these is least likely to be true? Um, Inhibition of protein synthesis in the cytosol would limit the assembly of new viruses. Um, that's probably true, right? If they prevent protein synthesis, then the the viruses wouldn't be able to um so enveloped, I guess it needs a protein code. So in that case, it would inhibit
What were you saying for envelope?
that it has a protein coat, right?
Um, so the um oh, I guess okay. I guess you mean um kind of like the spike proteins of like COVID.
Yeah, right. Like the it's like a protein coat that surrounds
Yeah.
the material, so that if you inhibit that, then it would prevent or limit the assembly of these viruses.
Yeah. Yeah.
That's
So yeah, when they say the enveloped one, they mean, with that plasma membrane around it. Those are the
yeah,
type of viruses that we can get, animals can get infected by. Um, so okay, so yeah, they're saying that this enters the host cell and it causes it to synthesize new viral components. And then what is least likely to be true? Um, and yeah, you have the right idea with lysogenic and stuff. So yeah, let's see. Yeah, let's go one by one. So A says, yeah, what are your thoughts on A over here?
it's true, right? A is saying that if they prevent protein synthesis in the cytosol, then it would limit the assembly of these viruses. So yeah. B saying disruption of trafficking between the Golgi apparatus and cell surface would result in fewer production of viral glycoproteins. So that's also true, right? If you prevent the Golgi apparatus from packaging essentially these viral proteins, um, then they wouldn't be able to be pushed out of the cell to then infect um, other cells to make viral components.
Hmm?
Um, so ceasing inhibition of lysosomal trafficking would prevent new viruses from being released. So if you inhibit lysosomal trafficking, yeah, this one doesn't really make sense, right? So I guess lysosomal trafficking would be inwards of the cell, right? It has to do with then that, yeah, that would increase
Mm-hmm.
uh, the virus, right? So I guess C would be not true.
Mm-hmm.
Um, and then D as well, saying disrupting protein synthesis in the rough ER would inhibit the release. Well, yeah, it would inhibit the formation, then they wouldn't be released. So it would be C.
Yep. And I just want you to tell me what separates like protein synthesis in the cytosol to in the RER.
Yeah. So um, in the cytosol, it's through ribosomes.
Mm-hmm.
I, and then I guess the rough ER would be through like studded ribosomes on the endoplasmic reticulum. One that like those are sent to be I guess functioning outside of the cell, and then in the cytosol maybe within the cell, either back to the nucleus, like the transcription.
Yeah,
Um,
how would it know Would
Um,
yeah.
I think they're called like nuclear localization sequences or something.
Uh-huh.
Um, and then I guess I imagine there's the equivalent for a sequence to be sent out of the cell for a specific function.
Yeah, do you remember the signal hypothesis?
Yeah, it sounds familiar. I don't remember exactly.
So it's basic. So like regularly, protein synthesis would happen in this in the cytosol with the ribosome. Um, but and that means like as the mRNA's codon is read, tRNA comes down with an anticodon that is also attached to the appropriate amino acid. So that's how the translation happens. And if a specific sequence of amino acids is made,
Yeah.
uh, it tells the ribosomes to like halt the protein synthesis, and
Yeah.
the ribosome will go and attach to like, the receptor of the RER, rough endoplasmic reticulum. I mean, it's called rough because of those ribosomes. So it attaches to that and like um let's say this is like, it'll attach to that and it would go through or rather, so let's say this is, oh, that's a terrible drawing. Let's say this is, the mRNA that's being produced. But if the
Mhm.
signal sequence is read, then it pauses it and then it attaches to some channel or whatever here, and then it restarts that protein synthesis, except the protein synthesis gets like, well, synthesized exactly or right inside of that endoplasmic reticulum.
Okay, cool.
Yeah, signal sequence is what.
Hypothesis. Okay. And then yeah, okay. I'll look into that a little bit more.
Yep,
But I
yep,
remember I remember it briefly from content review stuff.
yep, yep.
Um, okay, cool. Um, okay, so Plasmodium falciparum is a protozoan parasite known to cause malaria, an illness characterized by fever, chills, and flu-like symptoms. Which of the following is the most likely reason why P. falciparum would most likely be classified as a eukaryotic organism? So I guess here we're just looking at something that distinguishes eukaryotes from prokaryotes. Um,
Oh yeah, let me erase what I wrote.
it's fine. So A is it is able to inherit genetic information from another individual. Um, no, I mean that's bacteria can do that as well, right? Through the conjugation, um, or even like transformation from the environment. It's like released from a dead bacteria, so it wouldn't be A. Um, it has a spliceosome mediated mechanism for intron splicing. Yeah, I mean that's pretty much what separates eukaryotes from prokaryotes is the ability to splice introns. Um, so I mean, I guess it would be B in that case.
Yeah, right. Um, because yeah, of introns, essentially. So perfect.
Okay, and then yeah, I guess cell wall, cell membrane, and
Yeah,
that could
cell
be
wall would support the idea that it's more of like a prokaryote. Um, Cell membrane, why wouldn't that work?
it could be viruses as well, right? Because they could have like a cell membrane surrounding it.
So, um, but it could also be because prokaryotes, in addition to a cell wall, also have cell membranes,
Yeah.
right? Like gram positive would have a one cell membrane and a really thick cell wall. Gram negative would have one cell membrane, a thin cell wall, and then another cell membrane. So cell membrane is not unique to, you know, eukaryotes or prokaryotes and other stuff. But yeah, it's pretty much the intron stuff. And also, prokaryotes can have polycistronic mRNA, so their mRNA could code for more than one like protein.
Yeah.
So, yep.
Okay.
Did you know that half of the people who ever existed died from malaria?
Wow. I didn't know that. That's a crazy stat.
It's a
Half
crazy.
the people that ever existed. That's insane.
Yep.
Wow.
Mhm.
That's pretty surprising. And we still don't have I guess, is there a vaccine? There's no vaccine for malaria, right?
Um, I
Maybe
guess
they have one now.
the protozoa. Oh yeah, yeah. There are medications, but since it's a parasite, I don't think it'll be, you know, um, something that you can vaccinate.
Yeah.
But yeah, there's quinine. So like sometimes people who drink like have you know like a gin and tonic?
Oh yeah,
Hmm.
it's like that glow in the dark.
Yeah.
What
Yeah.
quinine I think it's called.
Yep. Sorry, say that again.
Like something quinone. I
Yeah.
forget what. Yeah.
Yep. Yep. Yeah. So like basically the Brits, I mean like so the reason that I guess like the old world with Europe and the Middle East and Africa, they all knew of each other's existence. But I always was like, okay, why did the Europeans, were able to reach the New World, Western Hemisphere in whatever 1492, but weren't able to go really deep into Africa till like 300 years later.
Yeah.
The reason is because of malaria.
Wow.
Once they started to, you know, find a way to treat that,
Mhm.
they found a way to treat that with quinoline, I think, or quinone, um, which would uh since the structure of it is has a lot of conjugation, that's going to make something give off like a color,
Huh?
like called
Oh, okay. Yeah.
fluorescence. Uh,
Mhm.
but also because, it was like the British people who were in colonies in Africa and stuff like that, like, it's a way for them to get the quinine into their bodies, like, in the form of like a beverage or something.
In classic English fashion, they added some gin.
Yeah, yeah, yeah. I think yeah. It's also pretty interesting because of, I guess, the weather being so like shitty makes that, you know, that they're gonna colonize places that have nicer kind of climates.
Yeah. Cool. I didn't know that. That's a good fact.
Hmm.
Nice.
Yeah.
Um, Kidoki. Well, I guess what time is it? 3:56. You said you had to go at 4:30. You have another student after that.
Uh,
Um,
yeah. Just
you
stuff.
want to do a cars passage then?
Um, we
Or
could.
should
I wonder if we'll have enough.
or maybe we should just finish with this and we'll do cars next week.
Okay. Yep. Let's do that.
So assume a single-celled organism was discovered in water above 100 degrees Celsius. On closer inspection, it is determined that this organism is classified under the archaea domain. Given this information, this organism most likely has so archaea is they're similar to both eukaryotes and prokaryotes. Um, Something that's like slipped from my memory. I know that they don't have telomeres, and I think Um, I guess if they don't have telomeres, then they most likely have a circular chromosome.
Yeah, that was perfect what you just did with how you deduce that.
Like,
Because yeah, if you don't have telomeres, which are at the ends of chromosomes, like why, right? Because if you get rid of like the telomere, wouldn't the part that's left be the new telomere?
yeah,
Yeah, circular chromosome. That was perfect how you reasoned that out.
it was like slowly adding things I knew. Uh, I don't remember exactly whether or not it had an endoplasmic reticulum or peptoglycan layer in its cell wall, but
Mm-hmm.
uh, I mean, I guess I'll submit it and then I'll review now. Let's see. Yeah, this is what I didn't remember. This part.
Yeah, so that seems like yeah, that seems like it would be a good thing to remember. So it seems like bacteria and archaea have the same everything except for one thing, which is the cell wall with peptidoglycan.
Okay. Yeah, I know that. So that's unique to bacteria then, and then archaea have everything but that, basically.
Yep, yep. So yeah, so that could be an easier way to remember. Just for everything, archaea is similar to bacteria except peptidoglycan cell wall.
Perfect.
Otherwise, it would be more similar to eukaryotes.
Yeah. One second, I'm gonna let my dog in.
Yep.
It's quite hot outside, so I think I'm too warm. I need to get in. Um, okay. Well, okay, this is a tough freestanding. Um, so two species of bacteria, Pseudomonas aeruginosa and Pseudomonas putida, were incubated separately for 24 hours in broth with or without the antibiotic ciprofloxacin. Afterward, a small portion of each group was transferred to fresh broth without the antibiotic, and bacterial growth was measured for another 24 hours, as shown in the graph below.
All right. What are
Which,
they telling us here?
sorry?
What are they telling us here?
So they're telling us we have two bacteria. That bacteria is sitting in a broth, I guess like a fluid with an antibiotic and without an antibiotic. And then after I guess it's placed in that broth for a day, then a sample of each group from each broth is transferred to a broth without antibiotic,
Yeah.
and basically they're just measuring bacterial growth
Hmm.
um, for another 24 hours, and that's what's on the graph.
Okay.
So
We see that um it seems like the Putida,
mhm.
um,
Seems like Butita.
seems to have the highest growth here. So I usually do like greater than signs and stuff, but it's easier here for me to just put like the number in, maybe like one, followed by the black circle two, followed
Yeah,
by the X three, and then four last. And it's really last. Like it's pretty much,
yeah, like no growth almost.
yeah. So it seems like ciprofloxacin is extremely effective for P. putida.
Yeah,
And what else do we see? We can also see that, um, oh, that, so what I labeled one, grown without antibiotic, that could possibly be some type of control of some sort.
yeah,
So that's like the full capacity of that. And the direct comparison would be, so, okay, so that actually helps us because this is, oh crap, these are different ones. Oh,
yeah. That's
one,
the
okay, I
the
meant
growth.
this. So yeah, this one would be the Putida without the antibiotic, and then this one, the Putida with the antibiotic.
Yeah,
And then the other one is the Aragonesa or whatever. I'll make that in green. So, oh, seems pretty similar, right?
Yeah, just one sort of grows faster than the other. I guess the one without antibiotics is growing faster.
Yeah. So all in all, what I would take away from this is that the Putida is tremendously, you know, affected by ciprofloxacin.
Yeah,
Yeah.
nice. All right, so we're just looking for something that is supported by the data.
Hmm.
So A saying treatment with no antibiotic results in greater rate of regrowth in aeruginosa and Butida. Um, so a greater rate of regrowth in both bacterias. Um, so I suppose the regrowth is saying like when they're transferring it over, right?
Hmm.
Um, great. And okay, so they're saying aeruginosa, the black circle has a greater rate of regrowth than Butida. No, we know that the Butida was the one that has the most growth.
Yeah, what are we comparing here for this answer choice?
I guess we're comparing rates, right?
Kind of, right? So, uh, yeah, so we're doing that, and what like things in the graph are we would we be looking for or comparing?
I guess um we're looking for growth number of bacterial cells per unit time, right?
Uh,
And seeing what has the fastest rate, or sorry, the greatest rate.
yeah, um so okay, so no antibiotic, so we're not going to be using well, what we are going to be using is this one, because that's the aeruginosa without the antibiotic.
Yeah.
And what's the other one we're gonna compare it to?
You know, the circle, the clear circle.
Yep, that's what we're comparing. Okay,
Yeah. Yeah.
so let's see. So the Putida grown without the antibiotic is going to be that,
Mhm.
and the Arganosa without the antibiotic will be that.
Yeah.
So which seems to have a greater, regrowth? Um, it's
The Butida for sure.
Yep,
So then A is wrong.
good.
Um, B is saying regrowth of aeruginosa treated with Cipro occurs more slowly than regrowth of Butida treated with Cipro. Um, so we're looking at the X and the square of Butida and aeruginosa.
Yep. So now we're comparing the ones with Cipro, right?
So we know that because Butida basically didn't grow, then aeruginosa definitely didn't occur slower.
Hmm.
Butida, so that's wrong as well.
Good.
C saying treatment with Cipro results in faster regrowth than treatment with no antibiotic and aeruginosa. Now we're looking at the X and the black circle.
Hmm.
Sorry. Yeah, Pseudomonas aeruginosa with and without Cipro. Um,
Yeah, so what are we comparing here for this one?
that we're comparing the X one, so Ruginosa with Cipro, and the black circle,
Good.
the without. So they're saying treatment with Cipro results in faster regrowth than no antibiotics, and we know that um that's not true.
Yeah, because they're kind of the same, right?
Yeah, pretty much.
Okay.
And then D, regrowth of aeruginosa but not Butida occurs more slowly after treatment with Cipro. So regrowth of adelinosa occurs more slowly after treatment with Cipro, but not Pipotida. Huh.
So what are we comparing here?
Here we're comparing both adelinosa and Putida with and without Cipro.
Um, with and without Cipro?
With Cipro.
Mm-hmm.
Um, well,
So,
they're
yep.
saying yeah, adelinosa and Pipotida. I'm just getting confused because they're saying the regrowth of adelinosa, which we know is occurring slower with Cipro, but so is Pipotida. Well, I guess Pipotida is not growing, so maybe that's it doesn't really occur slowly if it doesn't grow.
Mm-hmm.
I guess, right? That's the answer.
Mm-hmm.
Yeah. Yeah, I mean it makes sense. I guess really like I'm trying to it's a weird worded answer. So
Mm-hmm.
the regrowth of this bacteria of aeruginosa when it's treated with Cipro is slower. Um, I guess. I mean, I guess maybe the it starts to grow later, so maybe that's what they're saying is slower. But Futida doesn't really grow after it's treated with Cipro, right?
Yep.
Mhm. I guess. Yeah, yeah, it was so weird worded question, but
Yeah, when you say weird worded, do you mean things like rate of growth?
no, not those. I was talking about D. Like here, they're saying that the regrowth of this occurs slowly after treatment. Like we're comparing the ruginosa, but it sort of occurs. I mean, I guess at different points it grows at different speeds. Like here, basically the rate is a little faster than it is here.
Yeah, because you're looking at rate as slope, right? Yep.
Um,
So, yeah.
yeah, it was just confusing. And then like I guess this one, the Piputida is growing a little bit. You know, it does increase somewhat here. Um, yeah, I don't know. I guess I'm overthinking it.
Uh-huh. Let's see what they put down there. So they had, okay, I'll erase what I wrote. All right. So we have, yeah, I was gonna ask like what they did at that incubation step, which is just whatever, the binary fission kind of thing. Um, and
Yeah,
okay,
I guess they just put no regrowth there.
yeah. All right,
Nice.
cool. Let's look at another one.
All right. Okay. Scientists identify a new type of infectious agent that causes disease in humans. They hypothesize that this agent is a viroid. Which of the following observation best supports this hypothesis? So we're looking for something that characterizes a viroid, and that's sort of like it's basically like a virus without the protein shell. So it's really just like freestanding genetic material.
Yeah.
Um, so an agent surrounded by a protein coat that promotes entry into human cells. That's a virus, not a viroid. So it wouldn't support it.
Mhm.
The agent lacks genetic material but can still replicate inside human cells. No, the viroid is genetic material. C, the agent is found to be composed entirely of single circular RNA molecule. Yeah, that's probably, I mean, it could be sort of any form of genetic material.
Okay.
And then when exposed to the agent, endogenous human proteins become misfolded. That's a prion. So it would be C.
Yep. And viroids infect plants.
Yeah, that's what I remember. Yeah. Nice.
Well, I guess some of them could cause diseases in humans, like they wrote in the question stem. But yep, good job there.
Nice. Thank you.
Mhm.
Um, I guess hepatitis D is a viroid or a
Oh,
viroid.
yep. Okay, interesting.
Okay.
Yep.
So researchers isolated peptidoglycan from a eukaryotic cell. The best explanation for this would be that, well, we know peptidoglycan only exists in prokaryotes, so it's probably infected by um, a bacteria, maybe. So eukaryotic cell is a host to a bacterial species containing peptidoglycan. That's probably what it is. Because peptidoglycan, so for B, peptidoglycan is not a component of some cell walls. Membranes within the eukaryotic cell contain peptidoglycan as a structural element. No, that's like the same thing that they're saying here. D, a virus with protein coat contains peptidoglycan. A virus with a protein coat containing peptidoglycan infected the eukaryotic cell. Um, hmm. I mean, it's either A or D, but I'm leaning more towards A.
Mhm.
Um, because I know a virus can be like when it's enveloped, that could be a protein coat, but I don't know if it would have peptidoglycan. It'd be like glycoproteins or something.
Yeah, so in order for a virus to be able to infect animal cells, they must have a cell membrane, right?
Yeah.
And a cell membrane is composed of those phospholipid bilayer, right? So it shouldn't be a protein coat because sure, like the cell membrane could have proteins, transmembrane proteins, etc. But uh but yeah, when they say the protein coat here, they really mean like you know, that part that makes it like that icosahedral shape. Um, and also it's kind of like, but yeah, basically it would have to be a that's the only way that you can get that peptide glycan from, a eukaryotic cell to make sure that it's in there. Hmm.
Um, so a student analyzing dividing cells of a certain type under the microscope finds that each daughter cell produced contains half the number of chromosomes as its parent cell. Given this, cells of this type would most likely contain all the following except. So we're looking at cells, that dividing cells, where each daughter cell contains half the number of chromosomes as its parent cell. Um, so this is like meiosis, I guess. We're looking at something that is occurring during meiosis, right?
Um, let's see. Uh, well, I don't know if uh are you getting that from the part that says half?
Yeah, I guess. I mean, right, contains half the number of chromosomes as its parent cells.
Okay, good. So yeah, it could be the meiosis part, but yep.
Yeah. So it's likely to contain all the following except peroxisomes, um, dividing cells. I mean, yeah, I don't know how peroxisomes fit in here. I mean, they're like basically like, yeah, like break down matter or material inside the cell, right? Through oxidation or like free radicals, right?
Uh-huh. Yeah, yeah. Redox reactions And they particularly would break apart uh type of fats.
Mhm. Um, so I don't know how they would fall into this question. Um, so they're asking us dividing cells where the daughter cell contains half the number of chromosomes as its parent cell. Um, I mean, maybe, um, yeah, I don't know. I would skip A. I'm not sure how it relates. Maybe I can read through the other ones to see if I can get a better idea of what's going on in the question.
Yeah, you could also just so the question's asking, right? This is some type of dividing cell, blah blah blah, but what, would be not likely to find inside of this daughter cell? And there's no reason that peroxisomes wouldn't be inside of it, right?
Yeah.
Um,
I mean, I guess in that case, then we're looking at chromosomes have to be, I guess, we know that it has to be, well, I don't know if we know that it has to be an animal cell, but I'm assuming it's an animal cell. And in that case, it wouldn't have single-stranded genomes.
huh? Yeah,
Um,
right. Because you're gonna have the peroxisomes because there's nothing that tells you that you won't. Right? And nuclear pore proteins you're going to have. Microtubules, those make up, the cytoskeleton. So it's essentially just asking what would a cell not have? I mean, it
yeah,
not have, after whatever it's dividing, but yeah, single-stranded genomes shouldn't ever be there.
yeah, that makes sense. Nice. Okay, yeah, yeah, that makes sense.
Yeah. Yeah, these are really good visualizations. So like, yeah,
Yeah.
they're really quite good.
Apparently, they came out with a book recently, UWorld, like a book series.
Oh, interesting. I wonder if they have. Well, if it came out with it early or lately, then probably no one's, like scanned it and uploaded it yet for the rest of us.
I don't think so, but I'm sure someone will at some point.
Yeah.
Um, yeah, I'll look around to see. I mean, I have all the other books and stuff, but
Yeah, there's another one. So like the Berkeley Review, that's available. I have it in the shared Shane MCAT folder, but they made like for the new MCAT, like a psychology book, and I can't find any like scanned rips of that.
Mhm.
But I do have the book myself. But yeah, it would take too long to scan everything.
That's true. Is it a good book? Is that what you use for psych?
Yeah, yeah, yeah. I used in when it comes to books, like I used predominantly the Berkeley Review books.
Mhm.
And if there's something that I wanted to do more of a deep dive in, I would read like a textbook, like which I also have in the Shane MCAT folder. Like, there's a good textbook on biochem there, a good textbook on like anatomy and physiology, a good textbook on cell biology, good
Yeah.
textbook on physics. So yeah, I would use those like a textbook resource as well. Basically, I would use like lots of different resources, and in my notes, I would
Mhm.
it would be the definitive answer.
Okay.
Yep.
Okay, nice. Um, all right, I'll start doing that a bit more then if I can now that I have a bit more time, assuming.
Yeah, yeah, that makes sense. You're saying like writing out notes and stuff?
Yeah, like writing out notes or going to those textbooks and maybe like if I don't understand something well, then diving in a little bit deeper than just you rolled explanations.
Yeah, yeah. That being said, these explanations are pretty great.
Yeah, they're really solid. They're solid, and the pictures are great too as well.
Yep. Yep.
Um, nice. Okay, let's have time for another two maybe.
Hmm.
Okay, which of the following molecules is most likely found within the cell membranes of intestinal epithelial cells? Okay. I mean, off the bat, I know it's cellulose most likely. Um, or maybe not actually. No, sorry. I didn't look at peptidoglycan. Um, yeah, I mean, I guess that's only what's it called? Prokaryotes.
Well, what is it asking for here?
Oh, likely found. Oh, yeah, sorry, I didn't even read the question well. Most likely found within the cell membranes of intestinal epithelial cells. Okay. Um, so peptidoglycan we know is in prokaryotes. Um, B cholesterol. And
So
cholesterol inside.
would peptidoglycan be something that would be within the cell membranes of the intestinal epithelial cells?
No.
All right. So cross that out.
Um, cholesterol, would it be within the cell membrane? Um, not the membrane. I mean, like cholesterol is typically found in the bilipid layer, right? Um,
Isn't that the membrane though?
remember, I thought the um of epithelial cells isn't the membrane made up of like microfilaments, microtubules, intermediate filaments? Is that not the membrane?
So what you just mentioned make up the cytoskeleton,
Yeah.
right? Um, but the cell membrane is the phospholipid bilayer, right? And you could have transmembrane proteins there. You could have like proteins facing either end for, cell signaling and stuff. So, maybe let's think about cellulose. What's
Yeah.
that?
Cellulose is like the equivalent of glycogen, but in plants, right? Like,
Yeah, yeah, exactly what you just said. Yeah, it's just like yeah, so and it has so things that have beta glycosidic bonds, we don't have the enzyme to break that up, so we can't really eat grass and stuff like animals, certain animals do. So that shouldn't be it, right? And then cytoskeletal filaments, that's what makes up the cytoskeleton, aka like, you know, skeleton would be inside, right? Like our skeleton's inside our body. For a cell, it would be inside of the cell. So not sure if that would be something that would be in the cell membranes, but yes, it will be cholesterol. So cholesterol, like, kind of acts as a buffer in a way, like not chem buffer, but it can like either like let's say it gets too cold and the cell gets too rigid, the cholesterol can make it more fluid. And if the temperature is high and it's too fluid, the cholesterol will make it less fluid, more rigid. So yeah, so that's a big part of what you'll see if they ask you about cholesterol.
yeah,
Um, and that's also like what like the steroid hormones are made of, like testosterone and progesterone and estrogen and stuff. So, it's a four-ring structure. So yeah, it should be that.
yeah. I guess that's where I was getting confused because I was, yeah,
Yeah,
I guess,
glycolip. Oh yeah, yeah. So yeah, you see how that's all throughout like the inside? Yeah.
yeah. I probably feel like the cytoplasmic membrane.
Mhm.
That's not the cell membrane, right?
Yep, it is.
Oh, it is. It is the cell membrane.
Yep. So yeah,
Oh,
cytoplasmic membrane. I mean any membrane, right? Like especially if it's around. So like cytosolic membrane, plasma membrane, lipid bilayer, all that stuff would describe it.
okay. Okay, yeah. Yeah, I feel like Yeah, I don't know what happened there. I guess.
Oh, and
Uh,
then also like because there's these glycolipids,
mhm.
easy to mix that up with peptidoglycan.
Yeah, that's true. The glycolipids, yeah.
Yeah. But you can think
Yeah.
of it as peptidoglycan is like a protein sugar.
Mhm.
Glycolipid is a sugar like fat.
Okay, nice. All righty. Um, yeah, I guess I can finish these.
We could do one more.
Yeah. Okay. Um,
What was the one before this?
so
No, never mind.
I don't think there was other ones, right? Did I? Oh, I did skip them. Um,
Oh,
scrape.
sorry, yeah, go on.
Yeah, sorry. What were you gonna say about Scrapey?
Oh no, about the prions.
Oh yeah, I guess they're saying Scrapey is a transmissible neurodegenerative disease in sheep and goats caused by prions.
Yep.
Given this, a diagnosis of Scrapey can most likely be confirmed by, um, I mean, I guess we know that prions are proteins, so it's most likely Western blot, since that corresponds to protein.
Yep, that's pretty much it.
Yeah.
Um, what would siRNAs do?
sRNAs are they like I guess they're like they reduce the protein expression, right? Small interfering RNA.
Yep.
So I mean that wouldn't really tell us much, right? I guess maybe they would be maybe that would stop the prion from I don't know. Yeah.
Yeah, yeah. Yeah.
Stop it from attacking the cell or the misfolding the protein as much.
Yep. So, yeah, I think what they would say here is that it messes with the folding, like and tell you how to do the entire like whatever
Yeah.
process there. But yeah, if you go up just a little bit, I want to see. Okay, okay. Sorry. Where did they have the text?
Oh, sorry.
All right, right there, yeah. So, okay, here we go. So, have you ever heard of mad cow disease?
Yeah, Jacob Creutzfeldt disease, right? Is that what it is?
Oh, I didn't know that. But I just
Yeah.
remember when it was, yeah, what's up?
I know, I have heard of it. Yeah.
Yeah, so that's something that happened when they were feeding cows other cows, and there's prions will be like in your brain, and that's why it's so messed up. So in certain cultures where there's like cannibalism, they
Mhm.
there will also be this disease. They would call it, I forgot which, it's like a tribe, but they call it kuru.
Oh yeah. It's like in like New Zealand, right? Like close to New Zealand.
Yeah, I'm not sure, but you're probably right. Um, and yeah, there was like a show based on like the movie, Hannibal, like the serial killer guy. And so what he does in the show is like he cooks people and he gives them to other people to eat without them knowing it. And he does that with one person, like feeding him brains of a person, and that person, and then that character just like had really brain issues,
Right.
so yeah,
Yeah.
so that's the thing about prions, like they're going to be in your brain, which is, of course, where you don't want them to be. uh,
Yeah. Well,
uh
they're saying now that I
yep.
saw like read an article the other day about um I think it was like recombinant growth hormone. Well, not recombinant. Before they had it, they would like extract it from like pig
Oh
livers.
yeah, from their which gland was it or whatever. Yeah,
Uh,
for that. So like there's serostim, which is, yeah, so like, okay, so before something like this, to get growth hormones, we would have to take it from, I forgot which part of the brain. Anterior pituitary, flat peg.
Bacteria.
Yeah, the G in flat peg. So yeah, they would get it directly from animals. Um, but then they eventually made a synthetic version or a recombinant version that you mentioned called Serostem. So I used to work at an HIV like AIDS clinic, and yeah, it was a very good learning opportunity because most of the like all the patients, since they have HIV or AIDS, they have a host of other like comorbidities. So like each the patient would have like 20 diagnoses and like two, three medications for each, so like 60 meds and stuff. And one of the things that happens if you have HIV or AIDS is wasting syndrome, where your body just eats itself. So
Mhm.
we would prescribe and, administer serostim, which is a
Mhm.
recombinant growth hormone.
Nice.
What were you going to say about the recombinant growth hormone?
Oh no, I was just gonna say that not recombinant, but when they were taking it from animals, there was a prion in like one of the samples, and a bunch of people that they had given it to ended
Oh,
up with
yeah.
Alzheimer's at like 50 something years old.
Damn.
And they had found a link to like I guess like that prion to Alzheimer's
Hmm.
formation of the plaques or something. I don't know what it was, but yeah. But yeah, that's how I know prions well, just because I've like read about it before.
Yeah, yeah. I just checked it out. There's this paper from 2024 that
Yeah,
talks about damn. Wow, okay. Yeah, it is Crutchfield-Jacob disease. Cadaver.
oh, cadaver, that's what it was.
'Cause
Yeah,
I was thinking like as we were talking, like it's human growth hormone, but why would they take it from like an animal, right? But yeah, yeah, and it is in the pituitary. Um, so let's see. I guess the prions maybe made it into that
yeah,
because
I think that's what I'm saying. Like,
the pituitary, like that's in your brain, right? So if you extract growth hormone from your pituitary, you might get some prions in there with it.
yeah, yeah, I
So
think God they use recombinant growth hormone now.
yeah, yeah, damn, yeah, that's super interesting.
Yeah, that was cool.
Okay,
Uh,
so let's see. Yeah, so you're doing really well on all these, and um, let's see. Yeah, when would you want to meet next? Oh wait, no, we're on Thursday at 3 p.m. Eastern, right?
yeah, I guess we can do Thursday. I mean, I'm thinking now maybe we can just put it off till next week, assuming that I put the exam off till September.
Uh-huh.
Maybe we don't need to condense the sessions as much.
Okay.
Uh, if you think that it's necessary, then yeah. But say maybe on Thursday I end up doing the full length, and then we meet next Tuesday again to go over it, or maybe I've gone over it already, and then the stuff that I struggled with, then we can go over. Right?
Okay. So, and I talked to you about how when you're doing Blueprint or Altius full length, not to do their cars, right?
Yeah, you said to do the Jack Weston
Hmm.
car.
Yeah. Either that or, from your diagnostic tool, but yeah. Um, it'll probably be easier to just do the Jack Weston one because it's like 59 questions, it's set up right. So yeah, good luck on that.
Yeah.
Let me know how you do. Like, email me, let me know.
Yeah.
Uh, and oh yeah, and then I can put you in. So do you want me to cancel you for Thursday the
Yeah,
6th?
I guess let's cancel Thursday if
All
that's
right.
all right. Hopefully it doesn't mess up your schedule.
Yep. No, no,
Uh,
it won't. And
so that we'll do next Tuesday, same time, 2:30.
next Tuesday am I don't have availability, but I do have availability for every other day next week.
Or let's do Wednesday then.
All right. And want to meet at 2 p.m. Eastern.
Yep, let's do that. I'll just write it here on the calendar. So I have it there. So 2 p.m. Eastern. next Wednesday. It's the 12th.
Yes.
All right, sweet.
Uh-huh.
All right. Just looking at the calendar now. So I guess technically if I move it till the 15th, I have, was that 3 months? Is that 3 months? 1, 2, 3 months. Yeah.
Yeah, I'll have to edit this for you. I'll try to do that by the time we meet next. But as you whatever you do on each of these days, like you could just edit the cell. So
Yeah.
like, you're not going to be doing, yeah, if you go to June, the cell for Wednesday,
Mhm.
like, just replace it with what you do. So, that could be like, something like that.
Yeah.
Maybe something like, oops. Yeah. So you could adjust it and yeah, I'll try to have it ready for you before we meet next.
Awesome. Thank you so much.
Yep. Anything else?
Um, no, I guess like maybe for Psych Soc, I'll keep doing the Panko deck. Um, I've been doing like 45 minutes a day. I do the cards sort of slow. I feel like sometimes I do like 150 cards.
Uh-huh.
Uh, I mean, I imagine that's fine, right? I guess considering now that I have more time.
Yeah, yeah, yeah. As long as it's, just something that you do like every day, a little
Yeah.
bit every day. Um, and yeah, okay. So for that psych soc stuff, I have some notes in my note from my notebook that I put into that shared drive folder, like learning and memory. I have a bunch of scanned notes for that. So that could be
Awesome.
look at
Awesome.
as well. Yeah.
Okay, perfect. I'll look through that as well. Thank you, man.
Yep, of course. All right. Have a good week and weekend, and I'll see you on Wednesday, okay?
Take it easy.
All right, you too. So